Microbes have been discovered that can digest plastics at low temperatures. Scientists have found microbes in the Alps and the Arctic that could help solve the problem of plastic pollution. These microbes are able to digest plastic at low temperatures of only 15 degrees Celsius. The discovery could lead to a breakthrough in plastics recycling. The pollution of the Earth by plastics is one of the great problems of modern times. That is why scientists have been trying for years to find, grow or bioengineer microbes that can digest plastic. The search for and cultivation of similar microorganisms has become a new branch of business. So far, several species useful for this purpose have been found, but for their enzymes to work, high temperatures are needed, exceeding 30 degrees Celsius. The need to provide heat makes the whole process expensive and not carbon neutral. The search for microbes whose enzymes break down plastic also at low temperatures is underway all over the world. To find them, scientists from the Swiss Federal Institute, WSL, traveled to the high Alps and visited the polar regions. The expeditions turned out to be very promising. Microorganisms taken from alpine and Arctic soils were able to break down biodegradable plastics at 15 degrees Celsius, says lead study author Dr. Joel Ruthie from WSL. These organisms could help reduce the cost and environmental burden of enzymatic plastic recycling, he argues. The results and description of the new research have been published in the journal Frontiers in Microbiology. Ruthie and his colleagues sampled 19 strains of bacteria and 15 fungi growing on loose or intentionally buried plastic, which has been underground for a year. The samples came from Svalbard in Greenland and the Swiss Alps, specifically the Muot da Barberpida Peak, 2,979 meters above sea level. The isolated microbes were then grown in a dark laboratory at 15 degrees Celsius. The researchers identified them and found 13 types of bacteria from the groups of actinobacteria and proteobacteria, and 10 types of fungi from the groups of Ascomycota and Mucoromycota. The researchers then conducted tests to check the ability of each strain to digest sterile samples of non-biodegradable polyethylene, P, and biodegradable polyester and polyurethane, Per. The microbe's ability to digest two commercially available biodegradable blends of polybutylene adipate terephthalate, PBAT, and polylactic acid, PLA, was also tested. None of the species were able to digest P, even after 126 days of incubation. However, 56% strains, including 11 fungi and 8 bacteria, were able to digest Per at 15 degrees Celsius. 14 fungi and 3 bacteria degraded PBAT and PLA plastics. State-of-the-art tests confirmed that these strains were able to break down PBAT and PLA polymers into smaller particles. It was very surprising to us that a large proportion of the tested strains were able to degrade at least one of the tested plastics, says Ruthie. 2 uncharacterized fungal species from the genera Neodebresia and Lachnellula performed best. They were able to digest all tested plastics except P. The study results also showed that the ability to digest plastics depended on the culture medium, with each strain reacting differently to each of the 4 test media. How did microbes develop the ability to digest plastic? Since plastics have only been around since the 1950s, their ability to degrade was almost certainly not their original feature. Microbes produce many different enzymes that break down polymers. These substances are also involved in the decomposition of plant cell walls. Plant-damaging fungi are often excellent at biodegrading polyesters, all because of their ability to produce cutinases, which target plastic polymers due to their similarity to a plant polymer. Explains study co-author Dr. Beat Frey from WSL. Since Ruthie and his team tested the digestion of plastics only at 15 degrees Celsius. They do not yet know the optimal temperature at which the enzymes produced by bacteria and fungi will work. However, we know that most of them can grow between 4 and 20 degrees Celsius, with an optimum around 15 degrees Celsius, says Frey. A baby has been born in the UK with the DNA of 3 people. In the UK, a baby was born with genetic material from 3 people. All thanks to a method known as mitochondrial replacement therapy, MDT. This technique is an attempt to prevent the birth of children with devastating mitochondrial diseases. For several years, an experimental technique has been used in the world that allows the DNA of 3 people to be combined in one embryo. Thanks to it, newborn children can avoid rare genetic diseases. Now the body that controls all in vitro fertilization clinics in the UK, the Human Fertilization and Embryology Authority, HFEA, has confirmed the birth of fewer than 5 babies who carry the genetic code of 3 people. To protect the families' identities, authorities did not provide more details. In 2015, the UK became the first country in the world to pass legislation regulating methods to prevent women with damaged mitochondria from passing genetic defects to their children. The first child was born thanks to this technique in 2016 in the USA. Most of the genetic material is found in the nuclei of cells. However, about 0.1% DNA is found in the mitochondria, which are responsible for providing energy. They are inside the cell but outside the nucleus. This so-called mitochondrial DNA, mtDNA, it encodes only 37 genes, out of about 20,000 protein-coding genes. It is estimated that 1 in 6,000 children suffer from one of the diseases associated with mutations in the DNA. This applies to both nuclear and mitochondrial DNA. Mutations within mtDNA are mostly inherited from the mother, because the mother transmits mainly mitochondrial DNA. The risk of passing the mutation to offspring is very high. One scientist has found a way to prevent this. This is the MDT method mentioned earlier. So far, the Human Fertilization and Embryology Authority has approved 32 couples for treatment with this innovative method. In this method, scientists take genetic material from the egg or embryo of a woman with defective mitochondria. They then transfer the DNA to a donor egg or embryo with healthy mitochondria. Before that, they remove most of the key genetic information from it. The fertilized embryo is then transferred to the mother's womb. The genetic material from a donated egg is less than 1%. The entire DNA of the child treated in this way. A new method of treatment with mitochondrial donation gives families with a severe inherited disease the opportunity to have a healthy child, the HFEA statement reads. The agency said it was too early to give details of the treatments. Soon, however, we can expect a more extensive report on the treatment, which is being prepared by scientists from Newcastle University involved in the case. The UK requires any woman undergoing the procedure to obtain approval from the HFEA to qualify for the program. Families must have no other options to avoid passing on a genetic condition. The new method of treatment has many critics. They argue that there are other ways to avoid passing diseases on to your children, such as egg donation or screening. As they emphasize, Such experimental methods are not fully proven and may not be safe for children. Others warn that such manipulation of the genetic code is a simple way to design children at the parents' request. They may not only want to avoid hereditary diseases, but also have taller, stronger, smarter or better-looking offspring. Robin Lovell-Badge, a stem cell expert at the Francis Crick Institute, argues that it is now crucial to monitor the development of children who have DNA from 3 people. The possibility of observing the operation of the technique, mitochondrial donation, ED, on a practical level will be interesting. We will check whether the children are free from mitochondrial disease and whether there is any risk that they will develop any problems later in life, the scientist said. Cancers have accompanied man for millions of years. Cancer may seem like a disease that accompanies humanity recently. However, it is not. Scientists have discovered many prehistoric remains indicating that our ancestors suffered from cancer. The earliest documented case of the disease was discovered in the finger bone of an early human ancestor. Cancer was also written about thousands of years ago in medical texts. The first discovered case of cancer affected an individual of the species Paranthropus robustus, which lived about 1.7 million years ago. Our ancestor lived with a malignant tumor in the left finger bone. Archaeologists discovered the remains of his skeleton in the Swartkrans cave in South Africa. This area is often called the cradle of humanity, because there is the world's largest concentration of remains of human ancestors. Researchers compared CT scans of a finger bone fossil with images of modern cases of osteosarcoma. This is an aggressive form of cancer that attacks bone cells. The scientists' attention was drawn to the characteristic, cauliflower-like appearance of the bones. The research results were already described in 2016 in the South African Journal of Science. Currently, osteosarcoma is one of the most common bone cancers in humans and can occur at any age, although it most commonly affects children, teens and young adults. The American Cancer Society reports, the age of this prehistoric individual is unknown, but scientists believe it was an adult. In another paper also published in the South African Journal of Science, researchers described a benign tumor found in an individual that lived 1.9 million years ago. It belonged to the species Australopithecus sediba, and its remains were found in South Africa. It should come as no surprise that the first cases of cancer are discovered in fossilized bones, since other tissues have not had a chance to survive until now. Bone is one of the few tissues that can survive in the fossil record, explains Bruce Rothschild, a paleontologist at the Carnegie Museum of Natural History in Pittsburgh. However, even when disease remains are found in fossils, they are often invisible to the naked eye and further research is needed. About a third of cancers can be seen with the naked eye. X-ray images of fossils are needed to detect other changes inside the bones, explains Rothschild. The first written record of cancer dates back to around 3000 BC. It was around this time that an ancient Egyptian mathematician, physician, and architect named Imhotep wrote a papyrus manual on bodily injuries and surgical procedures. Today the text is known as the Edwin Smith Papyrus. Excerpts from it were published in the Cancer Letters. In total, Imhotep described 48 different medical cases in his work, including several related to breast cancer. The text was written in ancient Egyptian hieratic script. In it, Imhotep pointed out the characteristics of different types of tumors, including oily tumors and solid tumors. He also included descriptions of breast cancer, which he characterized as a bulging mass in the breast that spreads under the skin and is cool, hard and thick like the immature fruit of hemata. Imhotep gives a number of treatments for various diseases in the text. However, when describing a breast tumor, he laconically states that there is no therapy. A papyrus by an ancient Egyptian genius provides irrefutable evidence that thousands of years ago humans also struggled with cancer. It is also an invaluable source of knowledge about medical and surgical practices dating back thousands of years. We do not know how the fate of people with cancer from millions and thousands of years ago turned out. But like our ancestors, we are still looking for the causes of cancer and the best ways to treat it. Scientists prove that video games do not have a negative impact on mental health. Video games have become a popular form of entertainment in recent decades, arousing the interest of millions of players around the world. There has long been a debate about the impact of video games on the mental health of gamers. A study by Oxford University analyzed data from a survey of 40,000 gamers who participated in various video games including Animal Crossing, New Horizons, Apex Legends, EVE Online, Forza Horizon 4, Gran Turismo Sport, Outriders and The Crew 2. The results of the study showed no significant impact of video games on mental health, either positive or negative. Participants completed questionnaires and answered a series of questions related to their emotional well-being, social interactions and quality of life. The results of a study conducted by Oxford University showed no significant impact of video games on the mental health of participants. The study found that video games had neither positive nor negative effects on players' emotional and mental health. One of the key aspects of the study was examining the social interactions that occur in the context of video games. The results showed that video games can be a platform for social interaction and the creation of new connections between players. Some games, such as Animal Crossing, New Horizons, promote cooperation and mutual assistance between players, which can have a positive effect on their mental well-being. However, the study also found that intense gaming may lead to less time spent in actual social interactions, which may be a factor that needs further research. Apart from this fact, scientists have so far avoided the problem of loot boxes. Many politicians and public figures believe that loot boxes can be addictive. Despite the fact that there are many sites on the internet where, for example, you can get game upgrades for free, some players still spend a lot of money on in-game purchases. It should be noted that the Oxford University study has its limitations. Mental health issues are complex and multifaceted and can be influenced by other factors such as players' personal characteristics, social environment and lifestyle. In addition, survey data may have a degree of subjectivity and depend on participants' self-esteem. In exploring the icy moons of the solar system, we will be helped by autonomous robotic snakes. Engineers from the NASA Jet Propulsion Laboratory set themselves the goal of creating a robot that would be able to explore hard-to-reach areas, both on the surface and underwater. The result is a snake-like robot that could eventually be sent to the moons of Jupiter and Saturn to search for signs of life in the oceans hidden beneath the icy crust. The best place in the solar system to look for life, outside of Earth, of course, is on the moons of the two gas giants, Jupiter and Saturn. However, to study this type of objects, we need properly prepared probes that will have to be able to move efficiently in difficult environments. In their study, NASA engineers took the example of snakes. Such a structure could be a way to study both the surface and the inner ocean hidden under the icy crust. And such conditions occur on Europa or Enceladus, but also in other places. Drilling through a few to several kilometers thick ice crust can be problematic, especially on an alien world. On the other hand, such as Enceladus has geysers, as proved by the Cassini probe. They can connect directly to the subsurface ocean. A flexible robot that moves like a snake has a chance to get there, giving you the opportunity to explore this hard-to-reach environment. Previous attempts to search for life on other planets have shown us that we should not expect to find any traces of it on their surfaces. As an example, it suffices to cite the research that has been carried out on Mars so far. So if not on planetary surfaces, where in the solar system should we look for life? Interesting candidates here seem to be the ice satellites of the gas giants and potentially their oceans. However, studying them would be quite a challenge. First of all, we'd need something that could move underwater, but it doesn't end there. It would also be necessary to get to the inner ocean. Here we have different possibilities. To do this on Europa, a probe sent there would have to drill through kilometers of ice crust. On the other hand, e.g. on Enceladus, Saturn's moon, there are geysers that facilitate this task. But a probe wishing to use them would have to be characterized by appropriate resistance, as we do not know what the probe we sent would have to face on site. The best thing we can do is to prepare one that will be able to cope with various conditions. And it should be as narrow and as flexible as possible. In addition, we can direct it to the moon, on the surface of which we will find the presence of the most favorable cracks. As for the flexibility of the probe, the engineers decided that the best solution here would be to construct a robot that would move similarly to how snakes do. It has already been demonstrated by NASA's Jet Propulsion Laboratory, JPL, and is called the Exobiology Extant Life Surveyor, EELS. The first version weighs 100 kilograms and is 4 meters long. It is basically constantly improved after each attempt. And it needs to be properly programmed, because it will have to work autonomously. Even if we sent him instructions at the speed of light from Earth, for example to Enceladus, they would take at least 72 minutes to arrive. If EELS proved successful as a searcher for life on icy satellites, it would be all the more capable on the surface of our moon. And it would probably be able to explore it much more thoroughly, going where no previous probes have gone. It is definitely more versatile. It could find many uses on Earth as well, not only as a research tool, but also as a potential food supplier or even a lifesaver for people stuck in caves.